that material only you could study with X-ray and electrons. But in the case of light, like light can also be extended to another level called nanoscopy. So that's an interesting development which was awarded the Nobel Prize in 2014. So now we also have a nano, microorganisms. So we are not talking about microorganism. Now we have another level, organism which are smaller than micrometer. So we call them as nanoarchaea, nanoarchaeum. So we have another biology which is going to build up, like if you know that there are organisms which are smaller than micrometer scale. So we have something coming up in the field. Well, and that's not the only thing which has led to the development of this science, because microbiology is not just looking at the organism and then describing about the features, because we have something beyond that. So we were able to do that because we have two other ways of culturing and then preparing a pure culture. That has led into the large amount of data on microorganisms. So we have now a good field of research. So now we have several hypotheses which is related to human health and the ecosystem health, environmental health, public health. Everywhere we talk about microorganisms. So few days ago, some of you had some discussion about the importance of biological purity and genetic purity when you wanted to bring us sample for genomic studies. Okay, yesterday Dushyant, I think day before yesterday, he gave a beautiful talk how to avoid the contamination in your material, right? So but now we have real challenges in getting a material, because now the concept of microbiome is coming. So several organisms, which eukaryotic organisms which you and I understand are not individual organisms. They are actually meta-organisms. They have several microorganisms associated with it. So you need to know, like, what are you looking at? So it is not just one organism you are talking about. You are talking about a group of organisms. Those are meta-organisms. How do you talk about biological purity or genetic purity of a culture now? Okay, these are some of the difficulties when you go into eukaryotic system. Prokaryotic system are excellent system in a way that because we could get them in a pure culture. Also several challenges. There are several organisms which live together. So they will live in a dual culture. If you take them out, they won't be able to grow. Several methanogenic organisms, they live in association with the syntrophic organisms. So those organisms are difficult to purify. Several, sequencing projects finally, when they come to know that there are two different organisms, they didn't go further, they just stopped it, because they can't say that like they are working with a pure culture. So we now know about the scale of or the implication of these organisms on different scales, right? So now on Earth there are predictions that like we have over 10 to about 30 cells, microorganisms, and they do have a good reservoir of carbon, nitrogen and phosphorus. So beyond your like health perspective, like the diseases which are being produced by or caused by microorganisms, they have also an important role in nutrient cycling on the ecosystem services. Okay, so we need to know organisms, why do they do, how do they harvest energy from sunlight, or even without sunlight from UV rays, and all those things we need to understand, because we need to develop other techniques and technologies for applications and then to make use of those knowledge. So the challenge is also like they have a turnover time of 6 to 25 days. So whatever you are doing should be relevant, because those organisms are too fast to grow and then they replicate. Okay. So now that we have some understanding about the importance of microorganisms, and we should also know about like why do we have them in our laboratory. So the culture collection is important, because we need to have pure cultures so that we will be able to talk about the functions and physiological role played by a single culture, so that we will be able to say like, okay, this is the important organism to be studied or investigated. This was actually realized by Frank Call of Prague. He in 1911 he could have his own cultures, and that cultures he was able to sell it to other for a fee. And that's the way he started the culture. There is in fact a very beautiful paper by Malik and Klaas on bacterial culture collection, their importance to biotechnology and microbiology, which was published in the I think Biotechnology and Genetic Engineering Journal. Okay, that's an important. Maybe at this slide you can refer to that. It's an important paper which will give you the reason, like why we need to have culture collections. Okay. Well, when it comes to the importance is now realized. We have World Federation of Culture Collection. There is a organization which exclusively working on the culture collection information, and that's there in Utrecht in Netherlands. This group, this World Federation of Culture Collection is important organization in collaboration with the International Union for Biological Sciences and International Union for Microbiological Sciences. They have culture collection information. So you go to this website, you have information about the WDCM databases and also about the World Federation of Culture Collections. So why do we need to know about it? Because if you want to spend money, you should also know, are you going to spend on a new culture or a culture which has already been available or investigated. Otherwise you are going to repeat your efforts, like you are going to spend money on a culture for which the information is already available. So you need to have an idea like which are the cultures being studied or where we have information, and what is how to use that information for your own use. You need not spend so much money to sequence all the genome of a particular organism if you know that that is available, already available. Although you go to NCBI, but I think you also should know like whether they are available in authenticated culture collection of WFCC. Okay. So International Union of Microbiological Sciences is an important organization which has got 3 different domains. They have bacteria, bacteriology and applied microbiology (BAM), and also we have mycology and eukaryotic microbiology (MEM), and virology. So all of us can go to this website and then have an idea about like what are those cultures which are available in International Union of Microbiological Sciences. And if you go to WFCC website, there is a drop down menu where you can see WDCM databases. So they have information on CC info strains and WDCM reference strain catalog, and also global catalog of microorganisms, GCM. So this is an important website where from there you can get into the global catalog of microorganisms, and then you can go and then understand about like what are the organisms available. If you search, I think if you use this, you type the name of the organism which you wanted to know about, and then if you click search, and it will give you all the details about the data standards, overview, and about the features which are available about that particular organism. And also if you really want to know more about this particular website, there is a paper in BMC Genomics on global catalog of microorganisms, a comprehensive database and information retrieval, analysis, and then visualization system for microbial resources. That was published in 2013. So they have 2 modes of search. One is normal search, where you can refine and then also go by advanced search to know about like where do they actually isolated the culture, from which country, and all those informations are given in this website. And they have actually listed about 6 culture collections from India and their global catalog of microorganisms. So we have culture collections of Goa, and microbial culture collections from Chandigarh, and microbial research center from Pune, and then national fungal culture collections is also from Agarkar Research Institute, Pune, and national agriculturally important microbial culture collection from MAU and UP from ICR, and national repository for marine and cyanobacteria, microalgae and cyanobacteria from Bharathidasan University. So these are the ones, the cultures which are maintained here in these culture collections are reflected in global catalog of microorganisms. So these centers are important. Although individually we may have cultures, unless we deposit the cultures in these culture collection centers, they will not be represented in the global catalog of microorganisms. Right. So we have only few centers nationally which participate in this International Union of Microbiological Sciences, and they also list all the cultures which are available here. Well, So we also have, interestingly, some tools available in this website. WFCC has got tools. So the idea is databases do not come alone with the databases, but they also have analytical tools which you could employ in your studies. So these are also important. So you can visit this website and have an idea like what are the tools available for your own analysis. And I said about World Data Center for Microorganisms. So that having a culture collection, which is culture collection information worldwide. It's also another body. I'll just go to the next one to say about that. We have strains, CC info strains. That's what is known as CC info strains, means which are authenticated. It's a world directory of all registered culture collections. The database strain includes listing holdings of the registered culture collections. So whichever culture collection has been under the registered holdings, they will be given in this culture collections of WDCM. Okay, and just have an idea, now the number of bacteria is almost running into more than 1 million, 1.2 million culture collections which are available in WDCM. Right, and if you look at the way they started it, it was started by a Professor Skerman in Queensland of Australia in 1960. And from his laboratory, this went to Japan, and then it has now gone to China. So China, Professor Ma is actually taking care about all the culture collections, and so that's now available with the Chinese Professor Ma. Right. So we have also CC info strains, which has got several organisms listed, like we have bacteria, algae, and fungi, even several other things. If you go to this, you will be able to see all of them, right? And that World Data Center for Microorganisms is very, very important, because that is going to give you information about the cultures which are from the registered culture collection centers, right? And this is available with the but this website is also having an interesting drop down menu called WDCM SOP, standard operating procedures. Okay, so if you really want to know like what is being followed by them, you will be able to use it, because each one of us got a methodology, and if you don't have a standardized methodology, and even if you have a culture, if it is not if you are not using these methods, SOPs, then depositing them and then saying that your culture is new takes a lot of efforts, because they will screen out and say, okay, this is maybe a repeat or false positive, and then it's difficult to get into the WTCM, right? So this is all important for us, because as the registered culture collections, we don't appear in this WTCM. So there are about 46 globally registered culture collections, whose culture collections enter into WTCM, but from India we have none. So whatever we do, it's all like unauthenticated globally, but we do it because individually we have certain facilities in the laboratory, we do them, and we have culture collections. Well, we have not met the standards which are being laid out for getting into WTCM, right? And along with that, there is also another center called WFCC and Mersenne Center, that is Microbial Resources Centers Network. This is Mersenne Center is very common. Some of you must have come to know about these centers, because we have in Bhubaneswar, also in several places in India, in Mysore, CFTRI. So these centers are locally arranged resources center. Okay, thank you. And this also comes under WTCM. So these are important for us to know, because we can go into them to the website and also understand whether our culture which we are going to study are listed there, whether we have enough information about that. Couple of information about databases. If you look at this website, like we have CC info, which I talked about to you earlier, and also we have global catalog of microorganisms. These two I have talked about to you, because these are very important. And then there is something which is called a platform analyzer of bioresource citations. It's an interesting one for us, because that will give us several ideas about the culture which we would be able to use, because they have information about the papers, patent, genome, nucleotide sequences. So all at one go. So, if you are picking up a culture, if you want to really know what is known about it globally, you go into the analyzer of bioresource citations, ABC, they call ABC, and you will know everything about it. So, you need not do a lot of searching. Several of us, the point is you do not have to be under street light and then search again when it is already known where it is available. So, ABC is one such platform which will give you enough information about a particular culture, but those cultures are actually in CCInfo or GCM. So, these are two important things which you should know. If you have a new culture, well, you are a new hero in the field, because you got to know, you can write so many things about the new culture, but the culture is already available. You have a real challenge to say something new about it, right. Also, we have reference strain catalog. That's what I said when I said about the CCInfo strain before, and this is another word which they use, that is known as reference strain catalog, which is very important to us, because if you want to compare something, you need to have a reference to compare. So, you cannot compare with what? Like, you can't compare a bacterium with another eukaryotic system, because you need to know like whether one organism, a particular genus, and you want to compare with another one. Well, you can do that with the reference strain catalog. Okay. So, this got a few databases. This is what I explained to you: CCInfo, ABC, and GCM, and also WTCM reference strain catalog. And we got one thing which is about statistics on patent microorganism. That's a challenge. That's an interesting aspect about it, because if you really want to get you have a new culture, you want to patent it, well, you can go there and see whether it has been patented before, because you process, patent everything. Somebody comes, no, well, it's not possible at all now, because you need to know before whether it has been patented or enough information is available. So, you need to go to this website and see about what is available on patented microorganism before you do your process, you prepare for patenting. Well, and so we have now CCInfo, which has got about 7 or 8 culture collections from 72 countries and regions. So, it has got all the metadata. So, now the database, we use a word called metadata, because several databases from several collections. So, we use a word metadata. And also we have GCM, which has got having strain catalog information, and now we have about 368,000 strains from 103 culture collections in 43 countries and regions. So, the kind of information which you can get from this website is about on this many number of strains, 368 strains of different organisms are available. Well, we have ABC, which is a data mining tool. So, it is not a metadata, but it is about a tool extracting strain related publications, patents, nucleotide sequences, and then genome information from different public databases, form a knowledge base. And also we have reference strain catalog, and it maintains database of strains listed in an ISO, and also for the patented purposes. So, what do? They actually they go into CCInfo, and then they take out all the information from publication, patent, genome, JCI gold, and also nucleotide sequencing from NCBI. It's all going through the data mining tools of ABC. Then it get into the global catalog microorganisms. So, they have several screening procedures before it appears in GCM. So, that's a real tough to get something coming into this now, because we have so many organisms which are now available in GCM. So, if you are working with something, we need to know whether it's already available. So, you got to go there here and see. And GCM has got a different way of doing it. Again, similar process. They use primary sources like patent, journals, genome, nucleotide sequences. They call information crawling, and then go to regular expression like species name, remove duplication, false positive. Then they put it on a platform called SOLR, which is nothing but what they call is bioinformatics term for interaction with the web surface and then data provider. It's an open source enterprise. In a such platform it is available. What is important to know about it, like although I say many things, you have to refer to a paper which has been published in nucleic acid research. It's an important publication to know about GCM, right. So we have World Data Center for Microorganism, information on an information infrastructure to explore and utilize preserved microbial strains worldwide. This is about the preserved microbial resources. There are several organisms which are not cultured, not available in these culture collections, still you can work on them, right. And so world standard Data Center for Microorganism is also having a similar procedure to screen out and then to put it in the GCM or WTCM. So it's a real tough process to get any new organism listed in WTCM. Right, and if you look at the way the informations are being generated, you look at like US and Canada, and also from India, large extent from Australia. So we have several collections which are appearing now from these centers, and we have now almost 3 million microbials in the culture collections. So we have bacteria, fungi, and then virus. They also include cell lines. And if you look at the way India appears, it's almost now 5th in the rank of the which is having culture collections, which is about 2 lakh cultures which are maintained in a preserved manner. And what is more important is like good infrastructure and expertise in the long term preservation, characterization and identification of diverse group of microorganisms are necessary for a good service culture collections. So we need to raise our bar. Our laboratory should be in a position to come to that level of saying that we have cultures or institutes, or ICR for the matter. So we have one institute called NBIM in Mau, UP, which is doing the job, which is recently entered into these activities. So we never had it before. And we have MTCC and MCC, which is in Pune, is now almost they also, like I'll talk about another issue later on. So what is important is to have this structure, infrastructure and expertise. If they are not available, we won't be able to come to that level of acceptance by the international community. So if you look at the ABC and analyzer of bioresource citations, they talk about papers, patents, and then nucleotides and genomes information also. What is important is like there is a treaty called Budapest Treaty, where which is now been signed by several countries, almost 78 countries, administered by World Intellectual Property Organization in Geneva, and we are one of the signatories. And they have accepted the cultures deposited at any of the international depository authority to be recognized as a material which could be for the purpose of patent. So IDA is an important component, a feature of any culture collections. And if you look at it, like we have approved or rather recognized culture collection across the country. There are about 27. So I talked about Goa and then MTC, MCC, and the one national fungal culture collection in Agarkar Research Institute, and also MBI Mandal. But only 2 of them are recognized for the purpose of patent. So we have MTC, MTCC, and then MCC. So these 2 are the culture collections are recognized for the purpose of patents. Okay, and we can go to MTCC, which is IMTech. So there is one scientist, couple of research workers, and he is maintaining all the cultures which are sent across the country. And we have another one in National Center for Microbial Resource. They changed the name. Earlier it was National Center for Cell Science. It has now become National Center for Microbial Resources. So, but they don't accept only the cultures for patent purpose. They also have under general, safe, patent, and IDA deposit. So they have different fee structure for accepting the cultures. I tell you this story because as a microbiologist we would be interested to study, but if you like this is being already studied and then information is available, what do? We are repeating the efforts of someone else. You don't have to spend so much money in studying them, right? You can make use of that information for your further studies. We need to what you call change our strategy once we know that what is available outside, and then try to do something new. Well, MCC, what you call National Center for Microbial Resource Pune, has got a they have a procedure or they have a protocol wherein they talk about like how to deposit your culture under general, safe, and the patent purpose, and also for IDA purpose. I just wanted to show you this particular slide because It's not very clear anyhow, but they don't stop only with the genomic information, not only with the nucleotide information like 16S. No, they also go into MALDI, FAME, and DNA hybridization and all other stuff. So what are we now? I mean, like I want you to realize that, like, although we talk about genomic information, we talk only about the chemical communication of an organism using DNA alone, right? So DNA is the only chemical component which we are now talking about. That's an interesting mission statement of NCBI, which I will show you. So we leave out several other things. Organisms do not communicate with each other only with the DNA. They also do with protein and several other compounds, chemical compounds. The way I speak to you a particular language, and they do with the different languages. So we were bothered about only DNA genomic language, but several other things also we should take care about. Well, and well, I thought, like, let me also introduce you to one important component which your coordinator visited in somewhere in 2016 or 17, and this is known as Microbia. Although the Dutch, they say Microbia, it is Microbia, whatever you like to call it. It is now a museum of microorganisms. So it's in Amsterdam. So if you really want to know about the microorganism alive, get into this museum and have a feel of how microorganisms are on you, on several other living organisms. Okay. What is more important is, like, these in the website they say they took about 12 years to put the idea into implementation, and then they have what you call a building which actually they renovated, which was actually constructed in 1848. Just imagine, 148-year-old building, they renovated it, and then they are now having a museum on microorganism. So that building is also an important building, which is known as, I think, Ladder Loglen or something like that. Well, I am not able to read out this, but this website has got a beautiful photographs, and also you can visit the YouTube videos to know how microorganisms are available, which you could see in a museum rather. Well, this is about microorganism. Well, when we come to a microbiologist, if you ask me about a culture, I will say, first of all, if you want to really know authenticated, reliable source of microbial culture, I will say go to ATCC. ATCC is the one which has got the reputation now. Its headquarters in Manassas in Virginia, closer to NIH and Washington DC. So they have a beautiful website where they talk about microbial panels, and then resources for agriculture research, and bacteria, and several other things. Okay, they also have standards, services, and products. Those drop-down menu will give you what are the products available. They don't list, they don't limit with the microorganism. They also have cultures, culture collections, and seeds also are available now, and several other resources. They have media information, different media to be used for culturing organisms also available here. So when you go to AG Bio, agriculture research, they have a drop-down menu where they talk about phytopath. So you go into that, they have a long list of organisms which they have in their collections for you to use, and so we have these organisms which are available for purchase, like you can pay and get the cultures. So if you really want to know whether you are working with a particular organism, want to use some reference, go to ATCC. That's the way you can do that, and then you have to give a credit. And they have certified reference materials. So that means, like, they certified, okay, this is the material which we are giving. Go back to our the concept which he was talking about, Dr. Kumar, biological purity and genetic purity. You can be sure only when you have from an authenticated source. The one which you have in your laboratory may not be, I'll say, use the word of Dushan, salt, suspect. So you need to first suspect whether your culture is pure enough to work with. So these two are very important. ATCC does the job for you and for a payment. So you need to pay money to get the culture. Well, and also we have, there are several other websites which give you a lot of information about different organisms. I'll say about the algae. Algae has got a beautiful website. They have, this is based in Ireland, National University of Ireland. So this is algae based. They have, I could not read out the numbers because I don't want to memorize it. So I left it on the slide, but now you can just go there and see which is about 414 thousands of algae, okay, from different sources. So, although these organisms are like, you could see them, you do not need a microscope to see them. Sometimes yes, some of you say no, we need a microscope, but these algae are being collected from several sources, and they have two, I mean 2 of them have initiated this work. Now it is a global algal database for taxonomic nomenclature, and then distribution information is available in this. And we have one, it is in Manhattan, that is a fungal genetic stock center. So, again, so about the genetic purity of a fungal organism, which is very difficult to claim. So, we have lot of difficulty with the fungus, and then there is one stock center which is giving you enough information about the different groups of organisms. So, they have on a they have one particularly on Aspergillus homepage and Saccharomyces. These are of very interesting to us, like those of you who are working on Aspergillus can go into this to get a genetically pure cultures. Okay. And we have another one on E. coli, just on E. coli, nothing else. This is actually maintained by, I think this is in Gießen, University of Gießen, and they have a E. coli database collection. Again, they have a lot of other information about E. coli map, and then also like they have tools which are under different I mean like you could use from this website directly and to know about the E. coli. E. coli is the organism when you really want to do comparative analysis, comparative genomics, and this particular information is available in Gießen. And of course, coming to NCBI, which all of us are having great idea, but I will say that is not enough. You go to the website, probably visit regularly to know what is coming up every time, because I will say this is the forerunner, because they founded it in 1988, before we could think of, I mean anybody could think of the use of this information, and such a visionary statement they have. I really like this NCBI, but problem with the NCBI now is people have dumped so many nucleotide information which are not, I will say, genuine. So, now we have come to a problem of GI-GO. So, you must be knowing what computer scientists are saying about garbage in, garbage out. So, now NCBI has got a problem. So, although it is a meta database, I will say several mistakes are gone in. So, people are now slowly refining it, in the sense they take all the information from NCBI, create their own databases, and that is more reliable in several groups. So, NCBI is a big story. Well, I will stop with it because I do not want to sound very controversial, but still a good source for you, because they got beautiful way of teaching you or learning materials. Tools are there, tutorials are there, and of course, you can also have several literature downloaded. Some of you could not afford to buy book, go to this website, download books. Several books are available for downloading on genetics and genomics. So, if you really want to read, okay, you have now that like internet is free for you, download them, read them for your own understanding. Excellent website for several of them. And this guy has got a beautiful mission statement, from an alphabet of only 4 letters representing the chemical units of DNA emerges the syntax of life process, whose most complex expression is man. I do not know, they are limited with man, but several organisms. So, the language of focus in the field of molecular biology, that is a beautiful statement, and that is what, like the staggering volume of molecular data and its cryptic and subtle patterns have led to an absolute requirement computerized databases. So, we talk about computerized databases and analytical tools. This is a beautiful statement of NCBI. That is the reason you need to appreciate them, because they are still able to maintain that much of information, making it available to everybody who wants to use across the globe. Imagine if somebody says, no, I do not want to fund this source. Well, that is a doomsday for us. Okay. So, I talk about a couple of this website, you go there, we have genomes, genes, proteins and chemicals. So, you have drop down menus, talks about several issues which is very interesting to you. So, you can go into genome level, genes level and proteins. So we have several tools: GenBank, NCBI Bookshelf, BLAST, Entrance, Gene. So I'm not going to get into this because, well, that's going to take a lot of time for any one of us. So what do they do? The NCBI is still an important source for us, a metadata for us, because it doesn't limit only with the, and the projects or the information which is being gathered only from US, but they also actually coordinate with the ENA, European Nucleotide Archive, and DNA Data Bank of Japan, DDBJ. Okay. So they actually coordinate. So whatever information which is coming into the EMBL, ENA, EMBL, EBI, and then DDBJ of Japan, all of them get into NCBI, right. So we have, for that matter, now the amount of information is getting into petabytes. So it's nothing but 10 to the power 15. Okay. Now they are talking about having exabyte. It's I just wanted to put it in how many zeros, like 18 times 0 amount of information is going to be stored in NCBI by 2020. So just think about this kind of a computer they like to imply, store the information, supercomputer. So it's beyond our imagination. So people have to imagine that and create the facility, do all of them, and make it available to every one of us. That's a very tough job. And I also like to introduce you a couple of things about this, because now they have RNA submission wizard, which is a part of the NCBI portal, and it's a faster real-time analysis to assist the submitters on the RNA sequences from prokaryotes and eukaryotes. And now you can do it from using 16S, 23S, 16 and 23S intergenic spaces. Can also for eukaryotic samples, it can include both large and small subunit ribosomal RNA, nuclear RNA, and ITS regions, and internal transcribed spaces. And so whatever we now that he said, like most of the submissions are submitted using now Bankit. Okay. So we have now entrance system of NCBI has got it's a diverse set of 39 databases. It is not just only one database, got so many databases, and also about 22.5 billion records are available here. So if you look at it, if you have a sequence read archive runs into 4 million, and then genome information. So this is the important component of NCBI. Well, they have introduced Magic Blast, which is a command line tool, which can actually you can use an NGS data against a whole genome or transcriptome, which is also being now available. So these are few additions in the new version. Well, all these things are actually reported in the issue of 2008-18 nucleic acid research. So they have a paper, database resources of the National Center for Biotechnology Information in nucleic acid research. Okay, so you can have an idea what is being available in the new version of NCBI. Well, the competitor, another one, I will not say competitor, now they are all collaborators, EMBL, EBI. So this is European Molecular Biology Laboratory and European Bioinformatics Institute, which is now available in Cambridgeshire, what you call Wellcome Trust Genome Campus of Cambridge. So which is a very important database, so meta database sources for all those who are working in Europe. So we have a bias. People who work in US will submit to NCBI, those who are in Europe will submit to EMBL, and those who are in I will not say in Asia, but some of us will go to NCBI, but we don't go to Japan. But from people from Korea and some extent from China and other countries, Thailand, they submit all their information in DDBJ, the DNA Data Bank of Japan. Okay. So these are our own biases. So we have information. So you need to compare all of them. So as a researcher, you may not know what's going on in the European laboratory. If you do not know what is going on in Korea or in Japan laboratory, so you need to compare them. So now these three groups actually they coordinate, they put up all the information across in their own website, so that everyone can have an idea what is being submitted across. Okay. So EMBL and EBI has got several data resources, and then they have about 16 countries, which is very important. It's a open data, open source software, with several analytical tools and technical infrastructure. So they get from 16 different countries, and now in the last 2 years it has gone into 120 petabytes. So much of information is getting into this EMBL EBI. Matter because every day, people around 3.5 billion scientists actually they use the EMBL EBI site, and they got every week they have 38 million requests are being made to EMBL EBI website, and then can store about 155 petabytes, and so 10 to about 15 bytes of information from 64 countries. They are now being using. They are using this website. Okay. And they have a beautiful information on tools and data resources. So you can visit them and have an idea what is available as tools in EMBL EBI. Each tool has got a feature which is good for certain purposes. So something you can do in NCBI, come to EMBL for something else. You need to do it because each one has got different application purposes. Okay. So we have several data resources are also given here in EBI website. Well, and so we have you have procedures for how to submit your data and how the data is being made use. All those things are clearly given. So we have tutorials to see them. So you need to go through that before understanding what you can do with this website also, right? As I said to you, now they have 120 petabytes, which is now going to increase to exabytes. So we need to have computer which are having a blend of fastest SSD solid state disk, and then fast access HD and normal HD and several other backup also they are doing it. So all those are important to have the information available across the world. Okay. And what is more for you, I'll say you should also visit what is going on as research in EMBL. So they have information called PhD program, and they have research topics which are listed here. So you can plan, and then if you like to do your PhD from EMBL EBI, it's a real challenge, right? So if you know how to use several of these softwares, you can gain confidence and send your proposal. Well, if you are successful, you can visit one of the top laboratories in the world to do your PhD, right? And also these opportunities are available for postdoctoral research. So if you what I say is, it's not the biology alone which is matter here. You need to know how to make use of the computers, computer languages. So you need to use these two technologies together, so that you could understand the problems, apply them, and then you will be able to make use of these two for your own career growth. Well, also I give you a brief introduction about the DNA Data Bank of Japan, and they started somewhere in 1987, a year earlier than NCBI. In fact, they made the first release of the nucleotide sequences which are submitted in the National Institute of Genetics in Tsukuba in Japan. So from there it's a big database of all the nucleotide information going from Japan and from Korea and to some extent from Australia. Right. So they also have tools and sets and analytical samples, and then they have procedures like how to submit your data and sequencing and aligning data from next generation sequencing platforms, and they have functional genomics data and project samples. Some words will be common in some places. They use the word biosample, bioproject, and then in the submission of genome information and gene nucleotide, those things you have to be. You can go through this see from these websites. Okay. Well, as I said to you, they also have a similar challenges, now that you just have an idea how many bases of human organism is available, 10 to the power say 12. Okay. So it's somewhere around 10 to the power 12 bytes of information, bases information is available. Somewhere about E. coli, again it's about 10, 11. Okay. So much of information is available. So although we are all enthusiastic about using technologies, when you go outside and say, come on, so many bases, what do you do with that? So you So every one of us, when you add information, it's piling up, piling up, piling up. So there are some scientists who sit and then mine the data, come out with the concepts. That becomes a challenge now. That is actually going to happen in the future. So gathering, well, information is not a big challenge. So we have another organization, another internationally accepted center, which is called International Nucleotide Sequence Database Collaboration, which is a long-standing foundation initiative under DDBJ and EMBL, EBI and NCI. So they have put all the nucleotide information together, and that is known as International Nucleotide Sequence Database Collection. It's not talking about genome, but it is talking about the nucleotide. Okay. They also have another standard consortium, which is known as Genomics Standards Consortium. So now that we want to know, like, what you collect as a genome from a particular organism should have some standards. So we have a consortium for that, and that is Genomics Standard Consortium. So why need to bother about that? Because if you want to submit somewhere, they will come to verify all those things, whether you have followed some of the standard information, so that it is of some quality which could be put in the globally accepted website and databases. Otherwise, it will remain with you, right? So even in publications, they ask you whether you have followed these requirements before you submit. It's going to be really difficult in the future, because information is now it's sought on every level, right? So we have well, now that like I get into some of the specialized databases, which is I'll say DOE JCI. So they got a integrated microbial genomes and microbiomes. So they got IMG/M. So they have a website which talks about all the microorganisms. So they have annotated DNA and RNA sequence data of archaeal, bacterial, eukaryotic and viral genomes from cultured organisms, single cell genomes and genomes from metagenomes. So GFM, genomes from metagenomes, and also from uncultured archaea, bacteria and viruses, and metagenomes from environmental host associated engineered microbiome samples. So now this extending. So earlier concept of pure culture is now slowly widening to the accepting the concept of microbiome and uncultured organisms, and that is all available in IMGM of DOE JCI. Okay, so they got all the data sets related to single cell genomes, and also about the genomes from metagenomes and metagenomes from the environmental samples. We have a paper which is again published in Nucleic Acid Research in 2017 on integrated genome and metagenome comparative data analysis system, which gives you statistics about the number of genome which are being which you could refer or compare and study with this database, which is about 47,000. So we have a good amount of microorganisms whose genomes have been accepted and they are available, okay, in this website. Well, some of the details which you could also see from this paper. Okay, so they have 173 million protein coding genes. So when you look for some genes, it's going to be really a tough job, because you have so many genes which are available in several organisms, right? So we have another interesting database which is known as GOLD. GOLD is nothing but Genomes Online Database. So it is again maintained by JCI, which is in Walnut Creek of California. So they have several projects. There again they talk about biosample, bioproject, metagenome, anarchaea, bacteria, fungus, and all the stuff. So this is an important website where you can know more about the genome, right? And well, again I'll say GOLD. There is a beautiful paper in this year 2018 by Mukherjee in Nucleic Acid Research, where he talks about what is the data usage policy and why our input is critical to ensure high quality and accurate metadata in GOLD, and how to use them, like use the GOLD meta database for all your genomic analysis and comparative studies. And so we have the GOLD is organized in a 4-level classification system, where they talk about study, biosample, organism, and sequencing project and analysis project. So they have 4 level of classification. Study has got no limit to the number of sequencing analysis projects that can constitute a study. Biosample, it could be a sample or an organism, and sequencing project, so which talks about like whether you are going to do whole genome sequencing or targeted transcriptomes, targeted gene surveys, and several other things. And analysis, what do you do with that, like what kind of analysis project you are going to have, whether it is genome, metagenome, metatranscriptome, single gene screened, genome from metagenome, transcriptome, and combined assembly, and several other projects. So these are important to know. And where do they get all the information? I talked about 47,000 organisms whose genome are available in GOLD. They get most of the information from Genomic Encyclopedia of Bacteria and NAHEP, GBA. So this is a project which they are supporting, and also they have Genome 10,000K project. I think DBT is introducing similar 10,000 in our country. So if you are into this project, Earth Biogenome project, so these genome projects actually give you the standard data which could be put into their database. So if you are a member of these projects, well, your data is also of a good quality, then it can get into GOLD. So gold is real gold standard now to know about the genomes of microorganisms. Okay, and where they talk about complying with the minimum information about any sequence standards, MIXS. So this is now they are slowly introducing the criteria which could be which they would be looking for when you set the data for submission. So there are a couple of papers, and before going into that, I will talk about the sequencing project. So we have 215,000 sequencing projects in GOLD, and several others. Analysis projects is about 174,000 analysis projects. The scale of activity which is going across the globe, you can have an idea by the number of these projects in GOLD. Well, so I talked about the minimum standard. There are a couple of papers which talk about like how to set the standards. Like one is about the minimum information about a marker gene sequence, MIMAX, and the other one is MIXS, this minimum standard, minimum information about any sequence specification. This is again a paper which the paper from Nature Biotechnology in 2017. So which gives you about like what are the criteria which you should follow, or your data should meet, and so that they will get into the GOLD. And then they also have, there is another paper which is about the minimum information about a single amplified genome, MISAC, and metagenome assembled genome, that the minimum information about a metagenome assembled genome. So these two are, so we have different strategies of getting genome information. So the criteria, minimum information which they want before they accept your data in their database. The point is like what we do in the lab should have certain standards, otherwise your information is not going to get into the global repository. If you do not submit in the global repository, whatever you do is only I will say becomes a waste. So I may say that like it will just remain with you, will never be used by anybody else. So we need to know about these standards. Well, I have few databases to introduce. Those are about microbiomes online. This actually from I think it's from Jim. Well, I could not find further information on this, because some of them they disappear when they don't get sufficient funds to sustain. So that's a challenge. So I just wanted to bring to your knowledge, although some researchers they come out with a beautiful database, when the money or the funding is not there, not able to sustain it, database just simply closes on its own. Like this, I think this is one case where I thought, well, your reliance on a particular database cannot be permanent, cannot be for a long period. So you need to use different databases so that you know how to get your data or information from different databases rather than from single one. Maybe certain features are really good, but if they do not sustain it, you are in trouble. Okay, so we also like although I said about GOLD and IMG and M of the DOE JCI, and you also go into this website of Joint Genome Institute, and they have beautiful tools and several things which you could learn from them. So make use of this website. So do not rely only on database, also go there. Well, we have microbial genome database for comparative analysis. MBGD is a database for comparative analysis of completely sequenced microbial genomes, number of which is actually rapidly increasing. They have so functional category. So if you go there, and then we have menus, and you can also understand like they have different I mean, you can make your searches based on the function which you are looking at. So MBGD is an important database for us to do the comparative analysis. So I thought like this is one which you should also know about it, because RDB is an important project started by TG of Mexican State University, Michigan State University. So he was actually editor of AEM, and they have a beautiful project, ribosomal database project, and well, you have several functional gene pipeline repository. Those of us who are working with the microorganism want to know about the functions of different organisms. Go into this, the information about the gene sequences from different organisms are listed for several functions. So you can, if you want to design a new primer, if you want to test the primer which you are using, go to this website, and then you can design and then validate your primers. So this is an important project, although it didn't gain it's a small player compared to NCBI. So it is known only to people who are working with the gene level, and it's an important website for microbiologists. So they also use the minimum standard information for any sequence. They would now insist on that before taking your data and putting it in RDP. So this is an important database, and we have another database for fungal organism, which is known as FungiDB database, which is actually this is actually by eukaryotic pathogen genomics database resources of Pennsylvania, and now I think it has gone into University of Georgia. So this is those of us who are working with the fungal organism can use FungiDB for database. So we have eukaryotic pathogen genomics database resource of means a subsidiary is being actually seen in FungiDB. Another database collection is also available in Biosic, where you can go for the metabolic mapping functions, other things which you can understand. So they have 3 tier levels where they talk about human, yeast, and then EcoSig, which is a E. coli K12 database, and several other informations are available. So this is a collection of pathway and genome databases. So not only genome, also about the pathways which you could study in Biosys. So I'll just go through quickly because I think I'm running against time. So we have E. coli database. So this is the whole information about the E. coli strain K1 and K12, and then all other E. coli is also now available, K12, MG1655. So whatever you want to know about the genome, transcriptional regulation, transporters, metabolic pathways. So the information is now slowly getting into a single organism, but they don't limit only with the genomic information, but they are also getting into the what you call transporters, metabolic pathways, and transcriptional regulation and other things. So we have metabolic Metasec. It's a metabolic pathway database where you can make all the comparison about different metabolisms across in different organisms. So we have another interesting database which is called BacDive. It's a bacterial diversity database from Germany. Where you can you can understand about the bacterial diversity, and it's actually maintained by German culture collection, and they have information for 63,000 bacterial and archaeal strains, including 12,715 type strains. So all of them, they depend on the journal, International Journal of Systematic and Evolutionary Microbiology. So whatever information which gets published in this journal, they get into the BacDive. So any new organism you have to get into this, and then there are certain standards by which only they will accept it as a new organism, and that strains, whatever information you want to gather from those organisms, you can go into BacDive. And well, for pathologies, we have Phytopath, which is a database maintained by BBSRC of UK. So, you click and then about the pathogen, they have about 265 plus organisms on the on the fungal organisms, about 12 bacteria which they pathogenic organisms, also about the protists. It is an important database for all the pathologists who would like to know about the pathogens, and this is Phytopath. And we have also PhyPass, which is a pathogen host interaction, which is again maintained by BBSRC and Rothamsted International. So, UK websites, it is about the host interaction and pathogen interaction, which is available to you. Okay, I think I will say, before you launch your career in genomics, try to read these papers. Okay, these are very important, because this will give you an idea how sequencing is important, how sequencing is made, the rapid developments in sequencing project. Last day Dushyant was talking about, like, new techniques, new ideas being introduced that makes sequencing job cheaper and cheaper. Doesn't mean that everybody has to do everything sequenced, because he also gave an idea, like he convinced the one of those business people, and he said everybody who is suffering from diabetes or from cancer can be sequenced. That is a mad idea. Maybe that will happen in the future, but well, I am saying these developments are making things easier for us to sequence material. They have beautiful description about the events, the milestones in sequencing projects, and the recent developments in the techniques. Well, the whole talk I was running through one common information. This is about the database. Where do you get about the database? One is hit and trial. You go to Google, write something. If you get it, okay, that day you are lucky to get something. Well, that is a way. The other one is, every year nucleic acid research comes out with one volume on databases. So, first volume of the year, they have an issue which is about all the databases. So, you go and collect from 2009. So, you have every first issue or that volume, first year of year beginning. So, they have one particular issue on databases. That is what, like in the write up I gave, we have about 1550 databases or so many. So, everything is about the databases are being covered in the issue of nucleic acid research. Well, I also say I would like to take you to another society which is very important. So, when we have International Union of Microbiological Society, we should also have another union coming up. We have one which is about International Society for Biocuration. Okay. So, whatever data you have now, we have now to curate them. So, that is, we have a society for that is known as Biocuration Society. They also publish a journal called Database. So, now all your AG, CT, GCCT, everything can get into as a paper when you have certain standards in the database itself. It is an important development. So, even if you have data not able to decipher anything with that, send it to them. So, bank it, I will not say junk it. So, somebody will use it. Okay, use it. Well, I just want to give you this slide was interesting to me, because I thought I will introduce it to you, because whenever we get into a library, we have dilemma which book to pick up and read. Any subject you want to master, you get into the library, we do not know what to take. So, there again we will take up something which is important, then in that itself you have to do what is meaningful and what is not meaningful. So, leave them. So, same trouble with all of us, we will face the same trouble when you get into any database. So, they have so many things. So, you need to know what you need and then read that much, that is enough. Well, with that I will conclude my talk. Maybe we should also accept that this fact, that the more knowledge we gather, we are also more ignorant. Okay. Thank you.
All of you agree with me, that was really a fantastic presentation of various databases. When we approached Dr. Ramakrishnan, he readily agreed to deliver a lecture. Only thing we added is more workload on him. We wanted him to speak on databases for different cultures also. I think he justified his presentation. It was really a fantastic presentation. Let us say a big hand to Dr. Ramakrishnan.
Oh, thank you.
So, the core point he wanted to highlight here is the importance of culture collections and more so the importance of putting your culture in a safe place. That is a highlight, because you are all uploading your data in databases, for example nucleotide databases or protein databases, whatever it is. The putting the culture in some culture collection is as important as putting your data into the databases, because remember, data and the organism from where the data is being generated need to be preserved. Okay. With that, I thank Dr. Ramakrishnan profusely for accepting this invitation to deliver a talk, and I'll call upon Swami to propose a formal vote of thanks.
It's really a wonderful lecture given by Dr. Ramakrishnan sir. So thank you for accepting this for this lecture. And we are going to give you some tokens.
I used to give, now I'm receiving one. Thank you. Thank you very much. So if you have questions, you can ask me. Otherwise, you can go to the website. They will have answers for you. Okay. Anything?
Yeah, if you have any questions, you can ask. We have time.
Conflicts. Is there any case of conflicts between different databases?
Conflict will not, I mean, it's not being coming up, but of course, there are preferences among individual workers. Okay. So,
You ask your question in your mind, in the mind.
conflicts, no, I will tell, I will give, I got the answer. Okay, you can ask the question for the purpose of recording.
So many databases are there to has any conflict arose in the past or like in present regarding that which database is correct of any given organism.
I may not say which is correct, which is not wrong, but I will say preferences by individual scientists. I'll say in European workers, if you ask them, they will prefer EMBL, EBI database. So they know, they feel they whatever data going into the database are reliable. They may not, that's why I said like NCBI is now getting a reputation of GI-GO problem. Okay. And EMBL doesn't have, EMBL may have the problem, but well, they feel they don't have the problem. So it is, conflict will come up only when, well, we have a choice to remove the conflict or get away from it. So they do it by selecting certain databases, use it for their own work, leave out the other things. So we have choices of using and not using it. So conflict will come when there is a legal angle to that or some question about it. So if you have a new material, you want to really work on it, so you be sure that like what is reliable, you use it. Well, I will just extend your question to another level. So in all these days, we have been using 16S ribosomal RNA as a marker for identification of species, strains, or whatever it is. Now slowly, now it has become an issue that people like to use the whole genome information for naming a new species. So that means like people are now accept the problem. There are few issues related to those data of 16S ribosomal RNA. So that's not enough. So we will collect the whole genome information and move on to the next level. So conflict is now what do you call? It's a masked, and now the new standard has been set. So if you have a new organism, you have difficult time. Now onwards, it's going to be really difficult time for us to say anything new. So you really want to say something new, you have to get the whole genome done, and then you have to compare with the reference strain. Then you talk about submission as a new strain. Earlier days, by just sequencing 16S gene, you can talk about new entry and all. Now it's not accepted. So conflicts are masked, standards are increasing. So that's why I introduce you minimum standards. It's a word which they wanted to make sure that like whatever we do comes up to that level of standard, so that we don't create problem in making comparisons. Okay.
Any more questions from anybody?
Did you tell them, since I missed the initial part, did you tell them that there is a requirement of at least for patent purposes and all, you have to submit 2 recognized cultures when you want to, when you are submitting?
Actually, no,
it should be done in at least 2.
No, we need not do it in 2 places, madam. But we have 2 culture collections in India which are recognized as IDAs. International Depository Authority,
Yeah.
Deposit Authorities. So we have one in MTCC and another one is MCC of Pune, which is now known as National Resource, National Center for Microbial Resource. Okay, so you can do it in either places, but everything there is a payment. You need to pay a fee for putting it on a general deposit, saved deposit, and then for patent purpose again for International Deposit Authority. So fee increases along with that. So you go to the website of MRCC, I mean like Microbial Resource, Microbial Center for National Center for Microbial Resource, then you know about the fee structure. So it's a different fee structure. The point is like they will accept a culture only when they know, okay, you have certain issues with it, and you have to make sure that you got something new or something which can do some desired function which is better than the available one, then you will go into that. Otherwise, you can put it in the general mode and say, I mean, put it on the deposit.
Okay, just to extend the narration by Dr. Ramakrishnan, suppose if you want to put the cultures outside India, for example, he talked about German collection, he talked about American collection. So if you, anything which is going out of our country, please keep in your mind, you need to take permission from National Biodiversity Authority. So this is a mandatory. You cannot send your culture without taking permission from NBA. Okay, so maybe to avoid all those delays or kind of regulatory authority paperwork, he rightly put it. We have two centers in our country, one in Chandigarh under IMTech, another one in Pune, that is MCC. You can get the same weightage for your collection or culture just by depositing this culture within the country itself. So this is a provision. That's the reason why India is one of the signatory for IDA, and sir very beautifully put those statements. So you can deposit your cultures in IMTech or MCC. You will get the deposit number, and that has got a tag. That is, with that tag, it is internationally only portable tag, so anybody can access that information. Okay. So I think it was a beautiful talk, sir. I profusely compliment you once again. So with that, we'll conclude the second lecture scheduled this morning. Now we'll move into the students' presentation, right? So please, I hand over the mic to
How many we have? How much? Okay, we start with the students' presentations. We have 5 in line. So each will be given